Now, Th17 effector cells are critical in the response to extracellular bacteria. So as we've talked about, intracellular bacteria and extracellular bacteria need to be battled in different ways as they are located in different components of the host cells in the body. So type 3 adaptive immune responses to extracellular bacteria are dependent on Th17 cells. Th17 cells are effector cells that are generated from naive CD4 cells, which recognize viral peptides on MHC class II molecules. And naive, these are generated by the presence of IL-1, 23, TGF-beta, and IL-6. And as their innate type 3 ILC3s do, Th17 cells will produce IL-17 and IL-22, which will then activate neutrophils. Th17 cell responses to extracellular bacteria act in the following ways. So, as I've mentioned, IL-17 and 22 are produced by Th17 cells, and not only do these recruit neutrophils, but these two cytokines will also induce the production of antimicrobial peptides by local epithelial cells. So, the tissue in which the bacteria is replicating can be made more antimicrobial by the secretion of these peptides by the epithelial cells that contain that area. IL-22 that's produced by these Th17 cells also increases the amount that epithelial cells are turned over. And this is critical to prevent any sort of tissue damage that can be done due to the extracellular bacterial infection. And this also prevents bacteria from being colonized. So what I mean by that is if you, for example, had a gut infection or intestinal infection, increased epithelial turnover means that the bacteria can never sit on an intestinal wall as those cells are being turned over very rapidly. So this also prevents a very solid foundation for the bacteria for an infection. The IL-17 that's produced by Th17 cell will also activate stromal cells and myeloid cells to produce GCSF, which will stimulate neutrophil production in the bone marrow. As I've mentioned, circulating neutrophils are very critical in the immune response against bacteria, and having a large concentration of them is critical in maintaining a low level of bacterial burden. Now, neutrophils have a very short-lived innate life, and in order to get the most robust power from neutrophils, we need to have a lot of them to counteract that short life. And so as IL-17 is being produced, there's an increased amount of neutrophil production from the bone marrow. In addition, IL-17 activates stromal cells and epithelial cells to produce chemokines to recruit those neutrophils. So it's not enough to just have neutrophils being generated in the bone marrow. The neutrophils really need to know where to go. And IL-17 generates those chemokines to attract neutrophils to the site of infection. Th17 cells also produce CCL20, which is used as a chemoattractant for other Th17 cells so that we have lots and lots of Th17 cells at the site of infection, such that they can all perform these effector function to lead to the expulsion of extracellular bacteria. Now that we've gone through the T cell responses to bacteria, both intracellular and extracellular, let's dive into humoral immunity and complement. Now, humoral immunity against bacteria, or the generation of antibodies against bacteria, is very critical for a variety of different reasons. First, you can actually neutralize bacteria. So what this means is that for bacteria that tend to bind to the cell surfaces of their hosts via bacterial adhesions can lead to the internalization of bacteria, which will then allow for intracellular bacteria to propagate and replicate in their host cell. When these bacteria are covered in antibodies, the epitopes that typically interact with the receptors that allow for the bacterial uptake are no longer available. And so this prevents the presence of the antibodies on the cell surface of the bacteria, prevents the colonization and uptake of bacteria, which is very critical. And this is called neutralization. In addition, the presence of bacteria is critical in activating into leading to a more robust activation of more macrophages. So free immunoglobulins that are just swimming around in the blood or in the tissue can't cross-link the Fc receptors for which they're specific to activate macrophages. So here you can see that this free antibody here doesn't lead to the activation of the bacteria nor the destruction of the bacterium for which they're specific. However, on the cell surface of the bacterium, antibodies will bind in very close proximity, leading to aggregation of immunoglobulins on the bacterial cell surfaces. Then, when these antibody-coated bacteria interact with macrophages, this aggregation of immunoglobulins on their cell surface will lead to the cross-linking of the Fc receptors on the surface of the macrophages. This leads to macrophage activation, as well as leading to increased phagocytosis and destruction of the antibody-coated bacteria. Humoral immunity against bacteria is also very critical in inducing complement. So here you can see that within bloodstream, you can have various different small antigen antibody complexes that are within the circulation. And activation of complement leads to the deposition of these molecules, of many molecules of C3b of complement onto the immune complex. So you can see here we have this complex here that is then coated with C3b. Now, the complement receptor C3-1 on erythrocytes will bind to these grouped immune complexes via C3b, and then within the spleen and liver, phagocytic cells will remove these immune complexes from the erythrocyte surface and destroy these. Now, this is the very general concept of complement. However, this is also very critical in the clearance of bacterial infections. So here, for example, you could see that this bacterium is coated with various different complement factors, which also happens in the periphery, but also with levels of IgG antibodies that were generated by B cells that are specific for this bacterium. The presence of C3b and other complement components on the surface of bacterium and the presence of IgG both act in consortium to lead to very high levels of opsonization and destruction of this pathogen. As C3b, the complement component on the bacterium seen here, will bind to CR1, the receptor for which C3b is specific, and the antibody will simultaneously also bind to the Fc receptor for which it is specific. This bacteria will then be actively phagocytosed by the macrophage, and the macrophage membranes will then fuse, creating this membrane-enclosed vesicle called the phagosome around the phagocytosed bacterium. And then when lysosomes fuse with the phagosomes, these enzymes will then degrade the bacteria. So the presence of humoral immunity not only neutralizes the bacteria, but it also leads to their increased phagocytosis as complement and humoral immunity act together. Now, you might be thinking that it's kind of surprising that with all these immune responses against bacteria, that there's ever a bacterial infection that happens. Now, that could be surprising. However, bacteria and other pathogens are very tricky, and they evolve very quickly and rapidly to evade and subvert the immune defenses of the host. So we're going to be talking about multiple strategies that intracellular and extracellular bacteria used to subvert and evade the immune response of the host. So intracellular bacteria use a variety of different means to escape the host immune response. So, for example, they use antigenic variation. So antigenic variation is when they modulate what pili or fimbriae are expressed. And so this leads to antibodies that generally block the bacterial attachment becoming ineffective. So for example, if early on in an infection, a bacteria expresses a certain type of pili or a certain type of flagella, an adaptive immune response will be launched against that specific molecule. However, if the virus if the bacteria shifts what it is expressing, the adaptive immune response is no longer active to that bacteria, and the bacteria can then lead to increased propagation. In addition, intracellular bacteria will inhibit MAMP recognition and signaling. So MAMPs are associated molecular patterns, and the way that various different bacteria do this is, so for example, they can produce a peptidoglycan hydrolase, which blocks the detection of peptidoglycans by NODs. So that was one of the PRRs. So these bacteria here will prevent additional PAMP and PRR signaling from occurring. They also can secrete intracellular toxins, which block NF-kappa B and MAP kinase signaling pathways, preventing the initiation of an inflammatory immune response. In addition, bacteria can evolve resistance to antimicrobial peptides, either by degrading antimicrobial peptides directly, so AMPs here are antimicrobial peptides, or they'll modulate cell membrane phospholipids such that antimicrobial peptides are inactive and ineffective against the intracellular bacteria. In addition, intracellular bacteria are known for inhibiting the fusion of phagosomes with lysosomes. So many intracellular bacteria will actually live within phagosomes of cells because it's a very safe environment as long as they prevent lysosomes from fusing with the phagosome. That is when degradation of the bacteria happens, and they obviously do not want that to establish an infection. So many intracellular bacteria will actually release cell bacterial wall components, which will inhibit phagosomes from fusing with lysosomes to establish a chronic infection. In addition, many other bacteria have evolved actually to survive within the very harmful environment of the phagolysosome. So they've evolved cell walls that contain various different acids and other lipids that prevent them from being killed by lysosome enzymes. And also many intracellular bacteria have evolved to escape from phagosomes. So, for example, Listeria produces hemolysins, which will actually make the phagosome itself lyse, so that the bacteria can then live within the cytoplasm. Now, extracellular bacteria also have a variety of different mechanisms by which they subvert the immune responses of the host, including shielding or inhibition of PAMPs, as we talked about with intracellular bacteria. So here, bacteria have evolved ways of blocking the detection of LPS. So if you can remember, LPS is the outer wall component of gram-negative bacteria that is recognized by TLR4. And many gram-negative bacteria have evolved mechanisms to prevent the TLR4 LPS interaction. Again, you can antagonize TLR4 to prevent its activation. And you can also coat many bacteria have evolved mechanisms by which host proteins will cover their surface to prevent their activation by blocking the detection of their cell wall components, such as LPS or peptidoglycan. In addition, extracellular bacteria, as intracellular bacteria, also have this modulation of their antigens, which again prevents antibodies that are launched early infection or during a past infection, and it renders these antibodies inefficient or ineffective against the current infection. Extracellular bacteria have also evolved mechanisms of inhibiting opsonization. Now, opsonization is very critical, as you saw, in the removal of pathogens, particularly those that are coated with antibodies. So bacteria have evolved various different mechanisms to subvert this, including the cleavage of complement factors, so preventing complement from from settling onto the surface of bacteria. They also can block fixation of complement to prevent its signaling, and they can also prevent binding of antibodies to Fc receptors of phagocytes. Opsonization is ideal when it's working with complement and antibodies in consortium. So blocking both complement and Fc antibody interactions will both prevent opsonization. So other bacteria have evolved mechanisms to inhibit or scavenge reactive oxygen species. So reactive oxygen species are very critical in launching an immune response and maintaining inflammation. It's a signal of inflammation. So neutralizing reactive oxygen species by bacteria minimizes the signaling that the immune system is doing to tell the rest of the body that there is an infection. In addition, extracellular bacteria evolve resistance to antimicrobial peptides, as intracellular bacteria do. So, for example, they cleave antimicrobial peptides, and they also prevent the binding and functional insertion of antimicrobial peptides into the cell membrane. If you remember from an earlier slide where we talked about the mechanisms of antimicrobial peptides, they mostly function at the cell wall surface. So many extracellular bacteria have evolved mechanisms to prevent antimicrobial peptides from interacting with their cell surfaces. A more detailed example of immune evasion can be seen with Streptococcus. So there's various different types or subsets of strep, its nickname, and these actually differ based on their outside capsular polysaccharides. So you can see here that the actual foundation, represented by the red circle of strep, is the same, but different types of strep will have different types of polysaccharides on their surface. So when an individual is infected with one type of strep, an antibody infection or an antibody response to the infection will clear that infection. However, when a different type of strep infects that same individual, the antibodies that were generated in the first immune response cannot be active against this infection. So then you need to have another wave of adaptive immunity against the new strain. As adaptive immune responses take a couple weeks to be generated, this lag time between the infection to the generation of new antibodies allows for the establishment of an infection. Now, another way by which pathogens, or specifically bacteria, have evolved to evade immune responses is by actually modulating the host cells. So here's an example of how bacteria will inject effector molecules into their host cell. So bacteria use specialized secretion systems to inject effector molecules into host cells. So these are basically needle-like things that bacteria have on their surface, in which they take this needle, put it into the host cell, and this allows for a channel for these bacteria to take their effector molecules and put them into the target host directly, and this prevents a need for them to interact in close proximity. These nano-injectors tend to inject bacterial effector molecules that can either kill a target cell, it can neutralize it so that it prevents the cell from acting in an antimicrobial fashion, and can also modulate the immune response of the host. So those are just a few examples of how bacteria have evolved to evade and subvert the immune defenses of the host. So today we've gone through some basic bacteriology, including how to classify bacteria based on whether they're gram-positive and gram-negative. And this is critical because this allows for different PAMPs to be presented to the immune system to be recognized. And we also talked about the difference between intracellular and extracellular bacteria, as the immune responses to either of them should be different as they are different types of pathogens. We talked about some innate immune responses to bacteria, including antimicrobial peptides, PAMP recognition, and the role of phagocytes and ILCs in infection. We then talked about adaptive immune response, including different T cell effector functions to intracellular and extracellular bacteria, as well as the role of humoral immunity and complement in clearing of bacterial infections. And we concluded today with talking about how intracellular and extracellular bacteria evade immune defenses of the host. And that concludes our lecture on immunity to bacteria.